Okay, Supriya. So we are talking about the new topic, that is molecular basis of inheritance. Okay, and I given you a brief background just now that how this field, that is molecular biology, it is emerging as a new field, an upcoming field, wherein there are a lot of sophisticated techniques and what you can say softwares, instruments are involved, because of which you are able to get a lot of intricate details about the nucleic acids and their functioning within the cells with respect to their basic functioning, that is protein synthesis, the replication, their own regulation, how they are involved even in inheritance at the chemical level. And some part of it you have already learned in the lower classes with respect to biomolecules, the nucleic acid structures you have learned, right? And the basic idea about protein synthesis also you are having it, right?
Yeah.
So this lesson will take you on a tour to right from the beginning, okay, how these molecules were discovered. It was obviously not an overnight thing that somebody discovered this molecule and one fine day everything was clear to us, correct? It's a matter of many years and many scientists who have understood, who have explored, and with the advancement of sophisticated techniques we are able to have more clear data about all this. And this is something, the story is not over obviously, okay, like with respect to plants, animals, humans, microorganisms, the story will always continue and more discoveries will keep coming up. Okay, so people in different parts of the world, they are carrying out their own research, and because we are having bioinformatics also today, so everything is added to a common database where the analysis can also be possible, and huge sequences which is practically not possible to write manually or decide, it is to be done with the help of computer technology. So it's an amalgamation of everything where you will learn about molecular inheritance. Earlier you have learned about genetic inheritance and where you studied about the chromosomes also. Here we will go into more further details into it. Okay, so that's the background about this topic. Now let us begin with the main story. Okay. Okay, so let us start with something about how this discovery of the nucleic acids actually started. Okay, like how somebody came up with an idea, what are nucleic acids, where they are found, all that will be clearly understood. Okay? Yeah. See, it started somewhere in 1869. Okay. Before we go into the discovery of DNA, the actual thing at present you are aware that when you talk about DNA, okay, how is it located? We have a cell, okay, any cell, a living cell, where we have a nucleus, correct? Inside the nucleus, there is a chromatin material present, highly coiled. That chromatin material is made up of chromosomes, and chromosomes are made up of DNA. This is what we know at present on a very broad scale, correct? So here, inside the cell, inside the nucleus, where are these things actually located? In what form they are present, their chemical nature and everything. Let us take a look at that. So as far as this lesson is concerned, please remember this comes under something called general biology, okay, where it is not associated with only humans or plants or animals or something. This is something, one of the very foundation concepts of biology, which you will always have to study in detail. And here, whatever concepts which you are learning, these are very important concepts. So understand them very clearly, okay, so that it will help you in the future also, in whichever field of biological sciences you will go. Okay. So starting from the discovery of the DNA. Okay, what is the full form of DNA, Supriya?
Deoxyribonucleic acid.
Wonderful. So ये देखो DNA का जो discovery था, okay, it was not directly discovered as something called DNA first. ठीक है, 1869 की बात है. There was a person called Friedrich Mischer.
Mischer, yes.
Spelling ध्यान रखना, ठीक है? Friedrich Mischer. This person was working with WBCs, the white blood cells. Okay, you know something called pus?
Oh yes.
In circulation we have learned. whenever
Huh?
anybody suffers from any infection, okay,
Yes.
so from the wound that the person gets, there is a whitish or greenish colored discharge.
Huh?
That's a liquid called pus. Okay, so that pus is something which contains the dead WBCs which have fought against the microbes or the pathogens and the
Hmm.
dead pathogens also.
Yes, correct.
का एक मिश्रण होता है जिसको पस बोलते हैं, ठीक है? So these
Yes.
pus also contains cells of the
Hmm.
microbes as well as the WBCs. So what this
Yes.
person did, he in a local hospital, he
Hmm.
collected some bandages which were used for in patients. कि अगर किसी को
Oh.
infection होता है, okay,
Huh?
so on the site of the infection, the doctors put up a bandage.
Okay.
Basic पहले cleaning वगैरह करते हैं. कि suppose like if there is a person who meets with an accident on the road,
Hmm.
okay, if not a fatal accident or not a very serious one also, बस injury हुआ है suppose, तो
Huh?
हाथ पैर में चोट लगा है, खून निकल रहा है, कट हो गया, रैशेस हो गया, whatever, that person is
Yes.
supposed taken to the hospital. Okay,
Huh?
so on that wound जहाँ पे
हाँ.
मिट्टी वगैरह सब लगा रहेगा, डायरेक्टली बैंडेज
हाँ.
नहीं लगाते हैं. The first thing that is done is cleaning, and that is very important.
Cleaning. Yes.
Cleaning with a sterile solution. Clean वगैरह
Right.
करने के बाद, okay, then the
हाँ.
cotton is kept and the bandage is put to prevent
Yes.
further infection. Right?
हाँ.
Such bandages he collected from a local hospital which were used once, and उन
Yes.
bandages के ऊपर लगे हुए थे pus. So this person collected such bandages, okay, which was having the pus collected from patients. So what he
Yes.
did, सबसे पहले, like he handled it very carefully. Okay, what he
हाँ.
did, he used salt solution
Why would they use pus?
because that was the one containing cells, no? It was in 1869. उस टाइम पे यही उसको सूझा कि 1869 की बात है, ठीक है? That time what he did when he collected the bandages, so he first had to wash off the pus. So for that he used salt solution.
Okay.
Okay, so salt solution से wash करके he collected that liquid pus with the
Okay.
salt solution, and then to
Okay.
that he added a weak alkaline solution.
Okay.
Any alkaline solution, a weak one. So when this was added to it, what happened was the pus contained certain cells. See, a
Correct.
living cell was discovered in the 17th century, long time back. ये 1869 का बात है, तो at least people were aware that there is something called cell and nucleus.
Yes.
Cell was discovered by Robert Hooke and nucleus was discovered by Robert Brown.
Robots.
Okay.
Yes.
Okay, so that time he understood that because of the weak alkaline solution, the cells were lysed, that means broken.
Okay.
Cells
Broken.
broke and the nuclei were coming out of the cell.
Okay.
So
Got
in
it.
that solution, it contained the nuclei of the cells in the pus.
The pus.
Yes. So from that nuclei, he let the nuclei remain in that solution for some time. In that solution, he observed that from the nucleus, there is a unique chemical substance which was found to be present. Because it was obtained from the nucleus. That's why he simply called it as nuclein. उसको इतना समझा कि सेल के अंदर न्यूक्लियस था, सेल टूटे, न्यूक्लियस बाहर निकले, न्यूक्लियस में से there was some chemical which got isolated into the solution.
Okay. Got
What
it.
was the nature of the chemical? अणु वो बाद में पता चला. But he understood that this chemical is from the nucleus. So he gave the name as nuclein.
Nuclein. Okay, got it.
इस तरह के न्यूक्लिन को isolate करके this was analyzed chemically. What is the nuclein containing?
Okay.
It was found that it had very high amount of phosphorus and it also showed acidic properties.
Okay.
So because it was having phosphorus content, okay, it was phosphorus is one thing, okay? Now we know that it is from the phosphate group that is present, not just phosphorus.
हाँ.
And जो phosphate है, that is obtained by the dissociation of phosphoric acid, H3PO4. That means उसमें acidic properties है.
Wow, sir, your chemistry is
Okay.
nice.
Yeah, chemistry is good enough. So phosphoric acid में से acidic content आया, and it was isolated from the nucleus. इसीलिए this was given the name nucleic acid.
Nucleic acid. Oh, okay. Oh.
Because it was obtained from nucleus and it was having acidic properties.
It all came from a first.
Yes.
What?
Okay. Yes. So then later on it was confirmed that this nuclein which was isolated by mixture was a combination of proteins and nucleic acids both.
Okay, got it.
Both were present. And then later on it was discovered that DNA and RNA, these two are the nucleic acids in a cell. So this was the beginning of understanding of something called nucleic acids.
Okay,
From
got
us.
Wow.
Okay.
Okay, got it.
Okay.
Yes.
Then the next thing was like because of this discovery now, people got fascinated कि कुछ मिला है, नया चीज मिला. So let us see what is that. Let us explore more about it. So in the 1900s is that time, whatever technology was available, उसके use करके this study was carried out further.
Okay.
And there were experts who were called geneticists who study all this. Geneticists, molecular biologists, whatever you see. Okay. So more studies were carried out. People were also aware about something called chromosomes in the nucleus. Okay, so when this was understood कि ये जो substance है ये nucleus में से निकला हुआ है, so people were also aware about the fact that nucleus contained the chromosomes also. So then they combined both the facts and said कि chromosome में ही कुछ तो है, this substance. And then more study were carried out, and then it was understood that the chromosomes mainly composed of DNA and proteins, which we accept even today.
Yes.
Okay, so this DNA is our genetic material.
Okay.
Got it?
Oh.
Just a second.
Oh. Okay, got it.
Okay, so that's how we are understood that the genetic material is the DNA. इतना हमें समझ में आ गया. Okay, and then this fact was also understood that this DNA, which is a part of the chromosomes, they have a lot of role to play in inheritance and variation.
Yes.
Between different species. Between different species, the DNA and the proteins associated with it, they show a lot of differences. So this was the beginning. This was the beginning because of which people understood that this has got lots of scope to carry out research. There are millions of species with us. And इतना confirm हो गया कि cell के अंदर ये सारे nucleus and DNA proteins वगैरह. So why not study about all the species with respect to their DNA? And that gave rise to the beginning of molecular biology as one field.
Yes.
Getting it? Okay. Then earlier people were having, because of the lack of knowledge or lack of more studies, there were also wrong ideas about DNA which were known earlier.
Wrong idea?
Yes, wrong in the sense like earlier people thought that this DNA must be a small or a simple molecule.
Okay.
Later on it was understood it is not a small and simple one, it's a large molecule, a macromolecule, and having a very complicated structure. So more and more studies gave rise to these clearance about the DNA. Okay. And the shape, the charge, the function, the proteins, how they are connected with the DNA, that was understood later.
Okay,
so some group of experts also were of the opinion that proteins were the genetic material and not the DNA. So there were experiments which were carried out to confirm the fact that DNA genetic material hai ki proteins hai, or maybe something else. Okay, so like that many experiments were carried out. So there were about a lot of researches like this took place for about 25 years. In this period from 1928 to 1952. Okay, many people did lots of experiments and they gave rise to good conclusions. and then it was convinced that DNA was the genetic material and not the protein. So
Correct.
many such experiments were carried out, but some of the important experiments we are going to learn now.
Okay.
Their conclusion, how they were significant. Okay. So सबसे पहले, the first one was Griffith's experiments. Griffith. ठीक है?
Griffith.
This
Okay.
experiment was carried out in 1928, and
28.
he was a British medical officer called Frederick Griffith.
Oh, Frederick Griffith.
Yes.
सुना है
Suna. Okay. You're reacting as if he was your classmate. Okay.
मैंने.
So earlier one, जो पहले वाला first वाला जो बताया था, that was Friedrich Mischer,
Mr.
and this one is Frederick Griffith. Don't get confused.
Okay.
Okay. This person carried out a very interesting experiment, and he carried out his experiments on rats.
Rats.
Rats for experiment.
Wow.
Okay. Yes. So what did Griffith do? Simply, he worked on pneumonia disease. Pneumonia is a disease which is caused by a bacteria called Streptococcus pneumoniae. Pronounce kar. Well, Streptococcus.
Octopus.
Octopus nahi re.
Focus.
Pneumoniae. Haan.
I said it.
Yes, very good. So this bacteria causes pneumonia disease on humans as well as other mammals. Okay, but this bacteria has got two strains. One strain is called the S type.
S type.
Another strain is called the R type.
Okay.
Okay.
Yes.
S type stands for virulent. Matlab S type pathogen can cause the disease.
Oh.
R type does not cause the disease.
What
That's why
is
virulent?
the meaning of virulent?
Virulent means which can cause the disease. Non-virulent does not cause the disease.
Okay, got
And
it.
S type is a smooth strain है ना, इसलिए S type बोलते हैं. This bacteria produces a capsule around it. That's why it appears smooth.
Okay.
इसलिए encapsulated करके लिखा है.
Encapsulated. Oh, okay. Got it.
And
got
it is pathogenic. It can cause the disease.
Of course.
The other
Yes.
one is opposite. It is not a capsulated one. That is for rough and non-pathogenic, non-virulent.
That is easy.
Exactly
Okay,
two such types of strains of Streptococcus pneumoniae which Griffith used, and this bacteria causes the disease pneumonia. The S type causes disease.
Okay.
Okay. So this person Griffith carried out 4 experiments on this bacteria.
Oh no.
Okay, so directly we'll take a look at it from the flow chart or the diagram that is given. So what he did was, see, firstly, okay, सबसे पहले what he did, सबसे पहले what he did, he had taken rats for the experiment because rats are also known to suffer from pneumonia because of this bacteria.
Oh,
So
yes.
what did he do? First, he took a rat and he injected the rough strain bacteria into the rat.
Rough strain?
Rough strain, which is known to be a non-virulent strain.
Okay.
So what did Griffith do? He took a rough strain, uska culture, okay, he injected it onto a rat. So the expectation was it was a rough strain, non-virulent, so rat ko pneumonia nahi hona chahiye. And rat should not die.
No.
That's what exactly happened. The mouse remained alive.
Oh, yes.
Because of the R strain, like just expect kiya वैसे ही हुआ. Okay. Second experiment, what he did, another rat he took, उसमें डाला उसने smooth strain.
Okay.
Now smooth is known to be a virulent pathogenic. So the rat was expected to get pneumonia and die because of that. And that's what exactly happened. The mouse was dead.
Kill the rat.
Okay, relax. Listen, the mouse was dead. Okay.
Wow.
So 2 experiments, the same thing happened what he expected. Third one, what he did now, S strain, the smooth strain was known to be virulent.
Correct.
Okay, तो अगर directly smooth strain डाले तो rat got dead.
Yes.
So what he did, he took a smooth strain, but he
हाँ.
killed the strain. Heat killed it. Bacteria को.
समझा नहीं.
He took the S strain bacteria. He heated the bacteria, so the bacteria died.
Oh.
And then he injected the dead bacteria into the mouse.
Oh.
So again, what was
Then
expected?
he used rough.
Though it was a smooth strain, virulent. Okay, but उसको bacteria को मारने के बाद inject किया है. So it was expected the bacteria will not be able to infect the mouse, and the mouse should
And
remain.
that is how it
That
happened.
is what exactly happened. Third experiment
But
also
sir,
as expected. बोलो.
but अगर उसने उसको heat किया and the bacteria is dead, so now it is a rough stain.
No.
Correct.
Heat करने से rough कैसे हो जाएगा?
नहीं मतलब बैक्टीरिया चला गया तो नॉन वायरुलेंट हो गया वो.
मर गया ना बैक्टीरिया. नॉन वायरुलेंट भी जिंदा बैक्टीरिया है ना, दूसरा स्ट्रेन है वो.
ओके यार राइट.
मर गया मतलब वो स्मूथ ही है बट वो मर गया.
हाँ ओके
Got it?
got it. Yes.
Fourth experiment. See the fun. What he did, इसने heat killed smooth strain लिया. That means which is not able to cause any disease.
Yes.
And along with that, he mixed the rough strain without killing it.
Okay.
And ये जो दोनों बैक्टीरिया का जो मिक्सचर है, okay, heat killed smooth and rough strain which is alive, not a killed one. दोनों के मिक्सचर को इंजेक्ट किया तो ideally the rat should not have suffered from the disease.
Yes.
Because ये smooth strain मारा गया है, rough strain जिंदा है but non-virulent है. But in this case, what he saw that the rat, the mouse got pneumonia and it died, and that was very surprising.
Yes.
That was very surprising.
Yes.
But the reason was then it was understood. With later studies, it was understood that when the smooth strain bacteria was heat killed and then mixed with the rough strain, there was something which was transferred from the smooth strain to the rough strain, which made the rough strain from a non-virulent to a virulent one.
Oh.
So the rough strain bacteria got transformed into a pathogenic one.
Oh no. Okay.
ये जो phenomenon हुआ, and because of that the mouse died because of the rough strain. When it became pathogenic, it got pneumonia and it died. So rough strain का क्या हुआ? There was a very interesting phenomenon that happened called transformation. And this was the phenomenon which Griffith discovered. Okay, but exactly क्या transfer किया? Yes, exactly what was transferred from the smooth to the rough was not exactly known. Later on, from other experiments, it was confirmed. So there were two options, whether it was the DNA or the proteins.
Okay.
And from further experiments, it was confirmed it is the DNA which is the transforming principle.
Okay.
So Griffith के experiment का conclusion क्या निकलते हैं? DNA is the transforming principle.
Correct. Got it.
Transformation का meaning क्या हुआ? When the DNA gets transferred from one cell to another cell in a medium.
Correct.
Then it is called transformation.
Transformation.
So ये Griffith के experiment का बड़ा discovery था.
Okay.
Got it? Okay.
Yes. Got it.
Then the next experiment which was carried out in 1944. Griffith वाला किया गया था 1928 में. And in 1944, three people. Okay. They were microbiologists from the USA. Okay. Oswald Avery.
Oswald.
Avery.
Yeah.
Okay. Colin MacLeod. And
these people I know.
Macklin McCarthy.
Yes. Yeah.
Okay. These people I know means they are your neighbors.
No. वो लोग ने genetics में किया था.
Yes.
Material something.
So they were microbiologists at Rockefeller University in the New York.
Rockefeller.
Yes. Okay. ऐसे बड़े-बड़े universities में these researchers they carry out such experiments.
Okay, got it.
Griffith ने transformation discover किया था. But these three people, they gave the evidence to prove that the transforming principle is the DNA and the genetic material.
Yes, correct.
Okay.
Yes, I remember.
So what they did was, they had
They
three
did.
options: DNA, RNA, and proteins.
Yes.
They are known to occur inside the nucleus.
Hmm.
Okay. So what they did, from the cell-free extract of S strain, okay, and from
Viral.
the R cell. So S and R दोनों में से DNA, RNA, proteins, all three were extracted separately.
Oh.
Getting it?
Oh. Okay.
They were able to extract them separately. कि एक में से सिर्फ DNA निकाला, दूसरे में से सिर्फ proteins isolate किया, third में से RNA. Okay.
हाँ.
And then what they did, see, ये देखो. Okay. There are they use the enzymes. Now they used enzymes to degrade each one of them. जैसे there is an enzyme called protease.
Protease. Yes.
Protease क्या करता है? Proteins को degrade करता है.
हाँ.
Okay. And there is another enzyme called RNases.
RNases.
Which
Yeah,
degrade the
RNA
RNA molecule.
Yes.
So what they did, see, non-virulent R strain and heat-killed smooth strain.
Okay.
इन दोनों को mix किया and
Okay.
in two separate combinations. Yes.
And heat killed smooth virulent.
Okay.
क्या किया?
What they did, they took heat-killed smooth strain.
Okay.
And mixed with R strain. जैसे ग्रिफिथ ने किया था fourth experiment.
Yes.
Okay. To this combination, separately, they added three different enzymes.
Okay.
So to this combination, जब protease enzyme डाला गया, it was found that the mouse is dead.
3 mouse मार दिया.
इसका मीनिंग क्या हुआ? किसी protease enzyme के वजह से क्या होता है? Proteins get digested.
Correct.
So to this combination, when protease was added, it means the proteins in both the cells are digested. So if proteins are responsible for causing pneumonia, the mouse should have been alive. समझा? If proteins are responsible for causing pneumonia, the mouse should have been alive because protease के वजह से protein digest हो गया. But that did not happen. Okay. So by mixing with protease, he has proved that I have digested the proteins. So अगर proteins responsible है disease cause करने के लिए, तो with this enzyme, it should have been alive. But what was the observation that the mouse is dead? इसका मतलब क्या? Proteins is not transferred from S to the R strain.
Correct. Yes.
Got it? दूसरा set में क्या किया? RNase enzyme. There also it was found that the mouse was dead. That means RNA is not transformed.
Yes, correct.
And in the third case, there was another enzyme called DNase. DNase is actually a DNA digesting enzyme. So when this mixture of bacteria was treated with DNase, okay, the result was exactly opposite to that what Griffith got. The mouse remained alive.
Okay, got it.
Understood how they designed the experiment?
Yes,
so beautifully to confirm it. Like nobody has seen a protein molecule. Nobody has seen an RNA or a DNA molecule.
Yes.
But by imagination they designed कि ये enzyme डाला तो ये degrade हो जाएगा. So one by one we can try and confirm. So this was very nicely able to confirm that yes, what was transferred between the S and the R was nothing but DNA.
Okay, got it.
You understood this? And their confirmation was the transforming principle is DNA. But even after this wonderful experiment, there were some biologists who were not yet convinced. उनको ऐसा लग रहा था कि नहीं अभी भी कुछ तो गड़बड़ है, some chemical लोचा.
Yeah, of course.
Okay.
Yeah.
So see,
Huh?
Alfred Hershey and Martha
Hershey.
Chase in 1952.
Martha Chase.
Another experiment.
Yes.
Alfred Hershey and Martha Chase in 1952.
I know these
Okay.
people. Yeah.
What? Very good. Okay, so their experiment is known as the Hershey-Chase experiment. And for
Okay.
their experiment, what they used something called bacteriophage.
What is that now?
Bacteriophage is a virus that infects bacteria.
Infects bacteria.
Viruses are known to infect plants and animals, but there are certain viruses that can infect bacteria.
Oh no. Okay.
So they use this kind of pathogenic viruses on bacteria called bacteriophages. And along with that, they used radioactive phosphorus P32.
Sir, can you like explain?
Radioactive phosphorus P32 and radioactive sulfur S35.
What is that?
See, for any element of the periodic table, especially the ones with higher atomic weight, they have the property of having isotopes. In chemistry, you must have learned about it. You know what are isotopes? So every element has got its own isotopes.
Correct.
So some isotopes of certain elements, they have the property of emitting radiations.
Yes.
Okay, not all isotopes. So phosphorus ka normal atomic weight is 30.
Yes.
But another atom that is phosphorus with 32 is able to emit radioactivity.
Okay.
High energy radiation particles. You must have learned in physics and chemistry. Alpha, beta, gamma particles and all, right?
Yes.
It is about the neutron proton ratio. So we are not going to that.
Yes.
You know that the phosphorus with a higher atomic weight is able to emit radioactivity. Similarly, sulfur, whose normal atomic weight is 32, having another isotope with atomic weight 35, is also able to emit radioactivity.
Correct. Yes.
So the DNA and the proteins in the cell are having phosphorus and sulfur atoms in it.
Correct.
So what they did, they took a medium in which these bacteria were present. Okay. And in the medium, they added radioactive phosphorus and radioactive sulfur.
Okay. Okay.
Now see the fun. What happens? They are also using bacteriophage after this. Correct?
Okay.
Now, what they did, some of these viruses, the bacteriophage, they grow in a medium which contained radioactive phosphorus. And some other viruses, they have grown it in a medium that contained radioactive sulfur.
Okay.
But though ऐसे mediumly. In one medium, the radioactivity was only on phosphorus, not on sulfur. In the other one, radioactivity was on sulfur, not on phosphorus.
Correct.
Because phosphorus and sulfur are part of two different molecules in the cell.
Yes, correct.
So those viruses which were grown in a medium having radioactive phosphorus, their DNA was found to be radioactive. Because phosphorus is a part of DNA.
Okay.
And ये जो radioactivity होता है ना, we can actually trace where these radioactive elements go. There is a fluorescence and scopy available by which you can actually trace. So in the medium, मैंने phosphorus radioactive दिया था. But that phosphorus has gone into the DNA of the virus. Because when the virus multiplied in that medium, its DNA also multiplied. And for that multiplication of DNA, it has taken up the phosphorus from the medium.
Okay.
So
Okay. I got it.
Such a DNA is called as a labeled DNA.
Oh my God.
Because you can actually trace that.
Yes.
Okay. But in that same medium, the proteins which were formed were unlabeled. Because उसमें sulfur में radioactivity नहीं था.
नहीं.
So that indicates what? Phosphorus has gone into formation of DNA. Sulfur has gone into formation of proteins.
Okay, got it.
Confirm हो गया. So in the medium in which labeled DNA was produced, there was no proteins which were labeled. And in the other medium, it was the reverse, where the radioactive sulfur was provided. So proteins were labeled, DNA was unlabeled.
Unlabeled. Okay, got it.
Got it?
Got it. Yes.
Okay. So now
Yeah.
these two, now we have two sets of viruses, one with the labeled DNA, other with the labeled protein, and opposite ones are unlabeled.
Correct.
Now these radioactive bacteriophages, okay, they were made to infect another bacteria called E. coli. Bacteriophages are the viruses that infect bacteria. तो E. coli bacteria को इनसे infect कराया गया. Okay, and these E. coli bacteria, now preferably in such experiments, why E. coli is the most preferable one, most preferred bacteria? E. coli stands for Escherichia coli.
Escherichia coli.
E मतलब Escherichia, genus का नाम.
Yes.
Okay, now why is this bacteria the most preferred one for experiments? Because in case of bacteria, there is something called as generation time.
Generation time?
Yes. Generation time का मतलब है simply the amount of time required for the bacterial population to become double of itself.
Double?
Yes.
Okay, got it.
Bacteria living cells, unicellular ones.
Yes.
So if suppose a population of bacteria has got 100 cells, तो वो 100 cells का division होकर 200 होने में कितना time लगता है? That is called generation time.
Okay,
so, E. coli is one such bacteria which has got a very less generation time. That means population multiplies very fast.
Very fast, yes, correct.
Okay, and when the bacterial cell multiplies, it is like a simple mitotic division. Nucleus will divide, then the cell will divide.
Oh, हाँ,
तो
yes.
जब nucleus divide होगा तो उसके अंदर का DNA content भी divide होगा. Got it? Okay.
Yes.
Now, these radioactive bacteriophages were made to infect these E. coli bacteria.
E. coli bacteria.
और ये जो E. coli bacteria है, these bacteria were grown in a medium which did not have any radioactive phosphoryl sulfur. Normal medium.
Okay, okay, okay.
मतलब जो radioactivity है वो सिर्फ viruses में. Either DNA or the proteins.
Got it.
Now you understood? Okay. Now what happens is whenever I'll show you this way. If suppose this is a bacteria. If suppose this is a bacteria.
Okay.
Okay. There is the bacteriophage virus. Say like this. This virus. Consider, just एक बता रहा हूँ मैं ऐसे. Suppose this virus, bacteriophage, comes to the bacteria. Bacteria is having its own nuclear matter in the cytoplasm.
Okay.
जिसमें bacteria का खुद का DNA है.
हाँ.
And the virus is having its own DNA molecule.
Okay.
So when we say, okay, virus में कैसा होता है? It is having its own DNA molecule and which is surrounded by a protein coat.
Okay.
तो ये वायरस का डीएनए और ये वायरस का प्रोटीन और ये बैक्टीरिया का डीएनए. Remember this.
Okay, got it.
Now, when we say that this bacteriophage has infected the bacteria, तो वो क्या करते हैं? This will get attached to the bacterial cell wall first.
Yes.
And at the point of attachment, it will dissolve the cell wall.
Dissolve?
Yes. That part of the cell wall of the bacteria will be dissolved by the virus. And then virus का DNA bacteria के DNA के cell के अंदर डाल देगा. Okay, the DNA of the virus will be put inside the cytoplasm of the bacteria. So viral DNA is now inside.
It's being recorded. Oh, yeah.
Okay. So what I was telling that when this bacteriophage virus, it infects the bacteria. So the basic meaning of an infection is that this virus will attach to the bacterial cell. It will dissolve the cell wall at that point of contact, and the virus will put its DNA into the bacterial cell, and the bacterial DNA will get destroyed. Okay.
No.
And the bacterial cell machinery will be used by the viral DNA for its own multiplication. So there will be from one
Okay.
virus, many small virions will be produced. That's the
Virion.
meaning
Oh.
of an infection and why a virus infects a bacteria.
Mm.
Got it? So here you can
Okay,
see,
got
as I told you, the coating is made up of protein, which has got sulfur, and the DNA is the internal one that is transferred to the bacteria.
Correct.
And the DNA
Yes.
contains the phosphorus. So as we
How?
said, that there are two types of viruses we were having here. In one set
Yes.
of virus, the protein capsule was sulfur labeled,
Yes.
correct? And in the
Yes.
other one, it was a phosphorus labeled DNA code,
Correct.
correct? In both the cases, the virus infects the bacteria and transfers the DNA to the bacteria.
Correct. Yes.
And the viral protein coat does not enter the bacteria.
How? Oh, yes.
Only
You're
the
right.
DNA which
Yes.
goes inside, correct?
Yes.
So
Yes.
in the
Yes.
bacteria, in the bacterial cell, okay, which is growing on a normal medium, in one set, it was seen that there was no sulfur detected in the cells. The
This is which
radioactive sulfur was not detected in the bacterial cell,
sulfur?
but there was radioactive sulfur detected in the supernatant.
Supernet?
Supernatant. That means virus ने bacteria को infect किया. After infection, that solution was centrifuged, and the filtrate was taken as the supernatant.
Okay.
In that, there will be infected bacterial cells as well as the empty protein coat of the virus.
Okay. Got it.
Here, the protein coat was sulfur labeled. That's why the empty protein coat showed the supernatant, the sulfur content. But यहाँ पे क्या? The bacterial cell were showing radio labeled phosphorus. And supernatant में no phosphorus was detected. अभी protective phosphorus was a part of the DNA of the virus.
Yes.
But वो अभी कहाँ पे detect हुआ? Bacteria के DNA में.
Correct.
Which indicated what? DNA was the one which was transferred from the bacteriophage to the bacterium. तो ट्रांसफॉर्मेशन किसने किया? प्रोटीन ने या डीएनए ने? It was the DNA which was transformed.
Okay, got it. Transformation of the DNA.
Transformation took place because of DNA and not the protein.
Correct.
So using radioactivity and bacteriophages, this Harshi chase experiment also leads to the same conclusion, saying that DNA is the genetic material.
Yes, correct.
That was again proved by these experiments.
Yeah.
Now is it clear?
Yes, clear.
Okay, radioactivity के point of view समझा अच्छी तरह से?
Yes.
Okay, that was the outcome of this experiment.
Outcome.
Okay.
Yes, got it.
So here, based on these three experiments, what have we understood now? That the first thing, okay, first of all, it was understood how the genetic material or the nucleic acids were isolated from a nucleus of a cell.
Isolated, yes.
That was the very first beginning. If something is there in the nucleus which was found to have acidic properties, and that was present inside the nucleus of a cell. Then further studies were carried out. DNA क्या है, where it is located, how transformation takes place, and what is the one that is leading to transformation. So based on firstly by Griffith's experiment, secondly by MacLeod and McCarthy. Okay, that was carried out using 3 different enzymes. And third one was Hershey chase experiment, which used bacteriophage as well as radioactivity.
Okay, got it.
So based on 3 such experiments, it was concluded finally that the transforming principle is only the DNA. And what is transformation? The transfer of the DNA from one cell to another cell. Getting it? Okay,
Getting it.
so that was about
Yes.
transformation and how it takes place. That is the first part of the lesson. In the other
Yes.
part, now we have to see understanding about DNA is required, how it
Hmm.
functions and all. But the very first question that arises is such a huge molecule of DNA, how it is packed inside a small cell, a tiny one. So that is called as DNA packaging. Okay, how a huge molecule is packed inside. So in prokaryotes, it has got a different method. In eukaryotes, the method is, we will see both of them. Then we will discuss something about heterochromatin and euchromatin. Okay, followed by DNA replication. We have to learn how one DNA molecule gives rise to another one. Okay, followed by central dogma of life, that is protein synthesis.
Central dogma. Yes.
And DNA regulation by operon concept. And the last part of the lesson is very interesting, more of a theoretical type, you can say, that is about genomics. When you talk about DNA, you have a separate branch called genomics. Okay, what exactly is the genome?
Alag branch?
Yes. Okay, it's not about just one molecule of DNA, it's about all the DNA of a cell taken together.
Oh my god. Okay.
Genomics and how it is studied, what is the outcome, and then there are some other techniques like DNA fingerprinting वगैरह क्या करते हैं, that will be the end of the lesson. DNA fingerprinting वगैरह discuss करेंगे end में. Okay, so maybe you can say another 2-3 more terms and we will be able to wind up this lesson. Okay.
2-3. Oh wow.
Yes.
Okay.